Next, can I invite Dr. Luke Alderwick to upload his presentation, please? Luke is a senior lecturer at the University of Birmingham, and his research lab focuses on understanding the molecular basis of mycobacterial cell envelope biosynthesis. In addition, Luke is the director of the Birmingham Drug Discovery Facility, which enables him to collaborate extensively with academic drug discovery community, with the common goal of searching for new antibiotic therapies. His lab's currently funded by the BBSRC and MRC, and today he's going to talk about Azetidines kill Mycobacterium tuberculosis without detectable resistance by blocking mycolate biosynthesis. Over to you, Luke. Thank you.
Thanks for the introduction, John, and thank you for asking me to give a presentation today. Very much appreciate it. I want to do some acknowledgements first of all and explain that what I'm about to talk to you about today is very much a large collaborative effort between the University of Birmingham and John Fossey at the School of Chemistry. This is the work of one of my PhD students who's just actually submitted a thesis on Monday, Alice Lan. But it also involves collaborative support from Public Health England in the form of Joanna Bacon, as well as the Guangzhou Institute of Biomedical Health in China. Pockets of funding from the EPSRC Follow-on Fund, Wellcome Trust, which supported Alice, as well as the MRC Confidence in Concept. Also Simon Waddell from University of Sussex, we'll come on to later. I'm going to talk to you today about tuberculosis. I don't want to spend too much time on this slide. All I need to say really is that TB remains to be a global problem. We have got two epidemics really. One is coming under control, which is the kind of the drug sensitive strains of tuberculosis, but we have an increasing number of MDR, multi-drug resistant tuberculosis year on year, and we are in desperate need of new drugs. And that's as much as I'm going to say about that. So in this multi-collaborative effort between School of Chemistry, School of Biosciences and the Drug Discovery Facility, we screened a library, a small library, and found a number of hits from the laboratory of John Fossey. And these are essentially called the Azetidines. Now we screened a number of these, and I'm highlighting just 5 today because we actually screened anywhere upwards to around about sort of, I think it was approximately 90 different Azetidine analogues in the end. But I'm showing you the key ones here. This one here, BGAZ, stands for Birmingham Guangzhou Azetidine. We're going to call it Big As 1 just because. This was our first hit. We screened against Mycobacterium sphegmatis, BCG as the model organisms, through iterative rounds of kind of blinded, really sort of SAR. We arrived at this one here, which is our kind of our lead molecule at the moment, BGAZ, Big As 5. And all I want to say really is that these compounds are not just effective against model organisms, of course, but they are bactericidal against not only the laboratory strain of tuberculosis, but also clinical isolates, and specifically a multi-drug resistant clinical isolate, which is resistant to isoniazid and rifampicin, at a MIC of 2.8 micromoles, so not too shabby. Now, how do we go about finding mode of action in terms of mycobacteria? Well, the first thing you do is you try to generate mutants. So Alice spent months trying to generate mutants. She was able to identify 3, but only at low concentration of one of the azetidine compounds. Turns out that when you sequence the genomes of these mutants to try to identify the resistance conferring single nucleotide polymorphism, which is usually the way in which you identify the molecular target in mycobacteria, turns out that all the mutants are previously recorded background mutations within BCG. And so essentially these were kind of false positives. Essentially what we're talking about here are molecules, compounds, which demonstrate at the moment what's approaching zero frequency resistance. We are unable to generate resistant mutants, which specifically confer resistance in mutations away from these background mutations. So these are quite interesting molecules, and we'll come back to that later. So what is the mode of action? Well, we have to take very much a kind of classical microbiology approach where we first of all, investigated the kind of major macromolecular pathways by doing some labeling studies. And to cut a long story short, when you increase the concentration of the azetidines, it doesn't affect DNA, RNA, or protein biosynthesis. But what we saw is that we saw a loss of the synthesis of peptidoglycan, but also specifically lipids. And so this enabled us to kind of explore the cell envelope in further detail. Just to support this as well, a really cool assay from Joanna Bacon's lab in Public Health England. She's got a really cool flow cytometry assay where when you stain with Cytox green, which measures, of course, permeability across the membrane because it binds to DNA, and also calcium violet, which measures metabolic activity, looking at measuring cytoplasmic esterase activity. What we see is that we see a decrease in calcium violet at around about day 2, suggesting a reduction in cell viability, but then also an increase in Cytox green staining at day 6, which also leads to suggesting that it's increasing in cell permeability, possibly due to a cell wall defect. And this profile of these two azetidines actually looks a lot like that of isoniazid. And this assay has been previously reported and kind of led us to believe that actually something was happening in the cell envelope. With some support from Dr. Simon Waddell, we also did an RNA-seq transcriptomic profiling experiment where we treated Mycobacterium bovis BCG with 2 of our compounds, and then we obviously looked to see which genes were up and down regulated significantly. And so it turns out when you do a gene clustering analysis, there's further support of our hypothesis that these azetidines target aspects of cell envelope biosynthetic inhibition. We saw an upregulation of cell wall stress response genes. We saw an upregulation of sulfolipid and phospholipid biosynthesis, as well as detoxification pathways and also efflux pumps. What was interesting as well is we saw then a downregulation, a significant downregulation in lipid synthesis genes, genes involved in mycolic acid biosynthesis, and also the docile regulon. Okay, so a very sort of whistle-stop sort of tour of the biochemistry of the cell envelope. I've got to do this, unfortunately. What I want to focus on here are 2 key layers: the cell wall bound mycolates, denoted by these squiggly lines, and then the ones where the mycolates are attached to tree halos. So mycolates come in 2 forms: directly attached to the cell wall core, but also attached to tree halos, which enables for the incorporation into the cell wall core eventually. Now, so what do you do in order to investigate cell envelope lipids? Well, what you do is you culture mycobacteria, you then label, you treat with your azetidine molecules first of all, and then you label with C14 acetic acid to selectively label newly synthesized lipids, and then you can then extract using solvents and then separate by TLC and then compare to known standards. Okay, so what are we looking at here? Okay, well, the key thing here is highlighted in red are the extractable lipids that aren't directly attached to the cell wall core. And what you can see here is a selective reduction in TMM and TDM, which are these molecules here. And then when you do a slightly different approach where you do a pulse chase labeling experiment and you change the way in which you separate the TLCs, we can see then an accumulation of this thing called lipid X. But it turns out that lipid X is actually free mycolic acid, so free mycolate, not bound to either tree halos or the cell wall core. But when you look also then at the mycolates attached to the arabinogalactan and the cell wall core, which come from initially the ones here which are free, the TMM molecules, we see again a selective loss in the cell wall bound mycolic acids. Now, the fact that free mycolate was still being synthesized and released but not bound suggested that we're looking at some form of inhibition of late stage mycolate biosynthesis. We also then did some further pharmacokinetic and also toxicological profiling of our azetidine molecules. And it turns out that there's a certain sort of amount of variety in their physiochemical profile in terms of the hydrophobicity and hydrophilicity. But when you look at the profile, there's no discernible inhibition of the hERG, sodium potassium transporter, which is something that you really need to look at if you want to progress these molecules further, because this is usually kind of a go no go stage because of cardiotoxicity, as well as mitotoxicity in using HepG2 cells. It's within limits. And the other interesting thing as well is actually that these, specifically the big AS5, has got an extremely long half-life. So its existence systemically is very prolonged, suggesting we're going to get a therapeutic dose. Okay, so I only have 10 minutes here, so I'm going to wrap up pretty quickly. So what we've shown is that the Azetidines display potent antimycobacterial activity against MDR-TB with no detectable resistance. There's also no cross-resistance as well, which is something which is really important. I should also say that these compounds don't show any activity against gram-negative organisms, but they also show some good activity against gram-positive organisms, specifically MRSA. We've shown that these Azetidines inhibit late-stage mycolic acid biosynthesis, that they also show a suitable pharmacokinetic profile that does show promise for further development towards the clinic. What I haven't been able to talk to you about today or show you is that these Azetidines do target at least the antigen 85 complex, which are mycolate transferase enzymes involved in depositing mycolates onto the surface of the mycobacterial cell. And I could say you could take my word for it, but we have put this together in a preprint, and you can look at this at the bioarchive. So if you would like to look at this in more detail, please go ahead and look at this. This is to Alice Land, just to reiterate. This is primarily her work, along with Ishin from John Foss's lab in the School of Chemistry at Birmingham. And I'm going to stop there and thank you very much, and see if anyone else wants to ask me any questions.
Thank you very much, Luke.